Okay, well, let's get started today. It'll be, we've only got 3 lectures left, guys. Congratulations, we've made it to the end of the summer, and August is awfully close, isn't it? It's coming on really fast here. And we've all been looking forward to August since May. Right? So May and June and July, and we're finally here. And we've got one more exam to conquer. That'll be the final exam, and that's going to cover the urinary system, a little bit on acid and base regulation that the kidneys have a lot to do with, and then back to the reproductive systems. This week's our last lab. So next week we've got the lab exam on Tuesday, choice of 2 or 7 o'clock, and most of you will go to your regularly assigned time, if you will. But if you're normally in the afternoon, we'll expect to see you Tuesday of next week. If you're normally in the evening, we'll see you at 7 o'clock next week. And that exam will be over urinary and respiratory. And this exam in lecture will be over urinary, reproductive, and acid-base balance. This is going to be chapter 23, urinary system. I'll probably spend all of today and part of next time on this chapter. Chapter 24 will be the acid-base regulation. That's where we started the whole course. If you remember, we started off day 1 with chapter 24, water balance and electrolytes. And now we're going to go back and look at chapter 24 again in light of acid-base regulation and balance and how the kidneys are involved with that. And then we'll finish up with male and female reproductive system. Anything that I do not get to finish face-to-face with you, I will make, again, as bonus questions on the final exam. I don't know that there will be any, but if there are any, I'll put that as bonus. The last exam went well. I still have a couple of hybrid students that I have not graded their exams. That's my goal this evening, to have that done. But with the bonus points from the digestive system, scores were quite nice. And I was very pleased overall with the class performance. And those bonus points definitely came in to help you. I also posted your Connect points, as I mentioned, in an email. And basically what I did is I went into Connect, said how many points were possible from all the assignments, drew the line at 80%, which the syllabus says if you have 80% of all the available points, you'll get all 30 points in your grade book. If you got all 30 points, great, you're done. I'm not going to look again to make sure you did any more assignments. I recommend you do them. I think they're good to help you get ready for the final exam, but I'm not going to be looking again. If you got less than all 30 points, then by doing some of the last 4 Connect assignments, you can raise your score, and I will go back and reevaluate just those students who did not get all 30 points. So those points are in your bank total. Really all that's coming, lab reports, I know it's been a long time. Lab reports I should have finished by tonight, have those scores in, and so you've got your lab report at 30 points, your second case study, 25 points. You've got dropping the 4 lowest lecture quizzes, which I can't do until everybody's done next week. You have your 2 extra credit lecture quizzes, which are truly bonus extra credit points. Many of you have done those or are working on those. And then there'll be the last lab exam, the last lecture exam, and then there is a 5-point APR quiz that will help you get ready for the last lecture exam or lab exam. So there's still quite a few points to go into the point total for you, and still some very good point adjustments coming that should be positive for most of you. So that's just a little prelude on logistics. Any other questions that I'm forgetting about related to anything in the course going into the last week, last 10 days? Okay. I'm here. I've got 2 reports of strep throat so far. One from 106, one from 105. So wash your hands, right? This is not the time to get sick. So keep washing your hands, drink some fruit juice, stay healthy, please, please, please. 10 more days. 10 more days, you can collapse. I'll be collapsing, you can collapse, right? Well, we can all collapse together. But we've got to get through the next 10 days, and then we can take a little break that'll be well-deserved. So urinary system, starting up here. If any questions, let me know. You're going to be reviewing this anatomy in lab, so I'm not going to spend a lot of time on reviewing the anatomy slides 10 through 27. So look through those, and then when we get toward the end, also I'll have you review those on your end. Isn't this a beautiful little weird looking diagram? What is this thing? This blue-green thing is what? It's representing what? Yeah, the glomerulus, right? And the blue and the green, I have no idea what that means, but kind of cool. So just a really quick couple things. We know the urinary system's involved with ridding our body of waste products. From the beginning of anatomy and physiology, you're always told that, hey, our body is constantly making waste products. Our body is always doing cellular respiration, right? So cellular respiration is the idea that we take in sugar, we take in water, we take in oxygen, and from it we make CO2, which, as we discussed with the respiratory system, is an important waste product that we blow off as part of our respiratory system. But the kidney also has to deal with getting rid of some of that acid, and that's where we're kind of heading. We're going to talk about what happens to all that CO2. You know that a large percentage of your CO2 is dissolved in your plasma. If it's dissolved in your plasma, that means it can be gotten rid of through your kidneys. Right? So that's where we're going to be heading as far as that pH story goes. We're going to realize that as CO2 rises in your blood, some of that acid will be released through your kidneys. Okay? And the urinary system is, of course, closely connected to your reproductive systems, so that'll be part of our final exam. You'll hear it referred to sometimes as the urogenital system collectively, with the urinary and the genitals all connected as one, because they're coming from the same developmental tissues. So, again, we'll go over some of this anatomy in the lab. I'm not going to review this, so you'll hear about it in lab this week. This is the basic overall anatomy. As far as functions of the kidney, definitely we're eliminating wastes. We got that, right? We're getting rid of substances that are accumulating in our blood. That's part of our regular metabolism. We're also going to be regulating our blood volume and our blood pressure. So we'll come back and we'll talk about some cardiovascular ideas. We're also going to be talking about fluid levels and osmolarity, keeping those levels of your body, the interstitial fluid and your blood pressure, all of that regulated. And we're going to talk about how the kidneys make a hormone called renin or renin. You'll hear it pronounced both ways, renin or renin. And this is going to be an important enzyme in measuring and regulating your blood pressure. So we're not going to be able to get away from the cardiovascular system. We'll be talking about that quite a bit. We'll be talking also about the respiratory system. When we get to the urinary system, it comes at the end because it truly does unite so many of the functional systems. If you have a patient in ICU, what body systems are you most worried about? Breathing, heart rate, and kidney function. So these three go hand in hand, right? So their overall breathing, their overall respiratory and cardiovascular function are going to be critical to keeping your patient alive and well. So let's see how they all go together. In addition, another hormone that we've discussed in the past, erythropoietin or EPO, you'll see it abbreviated EPO. This is a hormone that is involved with red blood cell production. We're also going to see, as I've already alluded, that the kidneys as well as the respiratory system are going to be helping you keep your acid and base balance of your body. Remember, the pH of blood needs to be what? 7.4, right? That's going to be your norm. And the average, the normal average is what? 7.35 to 7.45. Right? So your average acceptable range of pH is going to be 7.35 to 7.45. If we don't keep it in that range, we're in trouble. We're also going to see that calcitriol is a hormone made in the kidney, which we've seen that hormone. It came in passing. We were talking about bone and calcium homeostasis. So the kidneys are involved with your bone production as well. And finally, we're going to see that in extreme starvation, nothing I suspect any of us have ever experienced, but in extreme starvation, the kidneys can contribute to gluconeogenesis. Now, what does that term mean to us? Genesis, the making of, neo, new, glucose. Right, because the brain needs glucose. The brain can't work on any other sugar molecule. And so if you truly, truly are in a state of extreme starvation, your body is able to break down protein to create new sugar, right, just to keep you alive. So in extreme situations, the kidney and the liver can do this thing called gluconeogenesis, making more sugar. So what sort of waste products are we dealing with here? What sort of waste products are we counting on the kidneys to get rid of? Metabolic waste, right? Waste products of our metabolism are going to include things like ammonia and urea. Now, I'm not going to ask you to know these chemical formulas, right? But you may recognize them. If you look at them, they're all very small, right? One of the unifying things here is they're all very small. And in fact, they're so small, they can do what? They can be filtered out of our blood through the glomerulus and into our urine, right? So remember, only small things are going to be able to get through the filtering barriers of the kidney. So number one, we've got ammonia and urea. Now these, the top ones, ammonia and urea, are breakdown products of your normal proteins. So as you're eating proteins, you are breaking them down into the amino acids, and from that you will get ammonia. And ammonia is a waste product. We can't let it accumulate in our body, and our body, the liver specifically, then further converts the ammonia into urea. Urea, urinary, uric, right? You'll hear this common theme go through this. There's also uric acid produced. Now, uric acid is from the catabolism of nucleic acids. Let's remember, what's catabolism? The breakdown, right? The breakdown of nucleic acids, things like DNA and RNA. Are you consuming DNA and RNA? It's not on our food label, but we're certainly consuming it, aren't we? Because every time we eat lettuce or anything we're eating muscle, right? We're eating protein, we're eating muscle of an animal, or we're eating vegetables or fruits, whatever it is, we're eating cells, and cells certainly have DNA and RNA. So we are consuming DNA and RNA in our diet. It doesn't give us any nutritional value, so we don't put it as calories or something on the nutrition label, but our body certainly has to take it in and break it down, and one of the products or byproducts of the catabolism of nucleic acids is uric acid. So we got uric acid, we have urea, we have ammonia. Those are all nitrogenous wastes because they all have nitrogen in them. So that's why they're called the nitrogenous wastes. They all have nitrogen in them. If you look carefully, nitrogen in the middle of ammonia, nitrogen on urea, right? You'll see nitrogen molecules in all of these. So that's why they're called the nitrogenous wastes. Another one is going to be creatine. Creatine is a product of creatine phosphate catabolism. Where did we talk about CP before? In muscle, right? The energy necessary for muscle to run. We talked about how there's that phosphogen system and the short-term and longer-term muscle molecules. So you know that the breakdown of creatine phosphate is important in your muscles. Again, there's a product here that we have to get rid of. There is clinically a measure called the BUN, the blood urea nitrogen level. And if a patient is having any kind of kidney issues, has had a kidney transplant, or you're worried about their urinary function, you'll be measuring their BUN levels. And what it is, it's a measure of how high are these nitrogenous wastes in your blood. You want to keep them relatively low, right? Because your kidney should normally be able to get rid of them. But if your kidney is not working properly, then these BUN levels will begin to rise, which can lead to toxicity and is an indication that your kidneys are not working well. So you'll hear about creatinine levels and BUN levels being measured oftentimes in patients with suspected or known kidney disorder. If you have elevated BUN levels, right, azotemia, and that is a clinical term meaning emia, meaning what? Condition of the blood, right? So azotemia is an elevated BUN, and as it says here, indicates that your kidneys are insufficient. They're not working sufficiently well. If overall you have uremia, right? Emia, condition of the blood. So if you have uremia, right, you have an increase in the blood of these uric acid-like molecules. So that would be an overall, again, a deficiency in your kidney function. But you could have uremia if you had, for example, diarrhea or vomiting or some other things. You might also see uremia. People who have uremia or have elevated BUN, really all we can do for them long-term is dialysis or organ transplant. So if they've got no kidney function, they need a kidney transplant, and until that transplant is available, we have to undergo dialysis or hemodialysis, same thing. Okay, filtering of the blood artificially. So what is the body doing? Or what are the kidneys doing specifically? They're getting rid of these waste products, but there's actually 4 body systems that work with the kidneys to get rid of waste products, and they're listed here. So we've already talked about the respiratory system in the past, right? We're getting rid of CO2. Remember, that's another way of thinking about we're getting rid of acid, because we know CO2 in water makes things more acidic. So to get rid of CO2 is to get rid of acid, and the respiratory system is hugely involved with that. We know that. The skin, we're sweating, and we do sweat out a small amount of urea through our sweat. So, okay, we get some of those waste products, not much, but we get some out through our sweat. The digestive system, we are definitely getting rid of some metabolic waste through our feces. And finally, the urinary system, our focus today, is getting rid of many of those nitrogenous wastes. So respiratory, integumentary, digestive, and urinary, all working together to get rid of waste products. So the next few slides are going to be just good old review. And I'll say a couple key words and move through these very quickly. Remember that the urinary system, the kidneys, and everything about them is retroperitoneal. That means that they are behind the peritoneum. That goes for the kidneys, that goes for the bladder, that goes for even the renal artery and the renal veins, and the adrenal glands. They would all be considered retroperitoneal. We know that the kidney has a hilum, that area into which everything leaves or enters the kidney. And the kidneys are not very well protected in the body as far as having a bony protective support around them, but they do have some fat layers around them. So the kidneys have a fibrous capsule, that's a tough connective tissue layer that's on the outside of the kidney. A lot of collagen in that. There's also quite a bit of fat going around that cushions the kidneys and keeps them in place and also protects them. And then there's a layer of tissue, very thin, that basically I think of it as being like shrink wrap or some sort of saran wrap, and it sort of binds the kidneys back to the dorsal side of the abdominal wall. So what's going to keep the kidneys in place? The fat and the renal fascia, and it's going to protect and keep the kidneys in their rightful place. Now within the kidney there is what's called the parenchyma. That's a word I have not used before. The parenchyma is the meaty glandular portion of the kidney, and we know that within the kidney there are all those pyramids, and this is the part of the kidney that's making the urine for us. If I look inside the kidney, if I look in the parenchyma or the functional part of the kidney, what I'll find is that there's a cortex, and we know the cortex, and we know that there's also a medulla. And the medulla you could further break down into pyramids, which are separated by columns. Right? So you see the pyramids within the medulla, and the structure, the tissue between the pyramids, is referred to as the columns. We have a variable number, 6 to 10, 6 to 15, you'll see different numbers, but a variable number of these pyramids in our kidneys. The collection side of it, where the urine is being collected, is going to go into minor calyces and major calyces, draining finally to the pelvis and to the ureter. So you'll be reviewing that in lab. As you look at a kidney like this, make sure you can label the basic structures. Again, we'll do this in lab today, but you have your pyramids. So here's a pyramid here, and two pyramids, and notice that they're separated by what's called the renal column. Okay, the renal columns. The outside is the renal, or the fibrous capsule, or the renal capsule. There is a cortex going around the outer layer, and then everything in the middle is the medulla with the pyramids and the columns. Urine is going to be collected into minor calyces, which will merge into major calyces, which will then all merge into a renal pelvis, which will then travel down the ureter. Kidneys are small compared to the amount of work that they do, but they receive a very large percent of your cardiac output. So somewhere around 21%, you'll read 20 to 25, but somewhere in that range, you've got a lot of blood flow going to your kidneys. And this is happening all the time. This is happening 24/7. So they're getting a very large portion of your blood flow. And that's important because we have to constantly get rid of these waste products. These waste products are being made 24/7 as well. So we don't get to take a break from our urinary waste production. The blood flow going into the kidney, we have the renal artery, which is then going to divide into segments. So the segmental arteries, the segmental arteries will then go into the interlobar, which means what? Interlobar means between the lobes. These are going to travel up the columns, if you will, between the pyramids. And then there are going to be the arcuate arteries. Arcuate, think arch. They're arching over the pyramids. And then there's the interlobular, which are going to go up into the cortex. Now, I never went through the individual names of those arteries in 105, so that's a new little bit extra for you to look at. Those also were not in the lab. They weren't mentioned in the lab as far as knowing those individually. Now, once we get through those 4 smaller segments, the segmental, the interlobar, the arcuate, and the interlobular, then we get to the afferent arterial, and that's where you should be more familiar with the story, picking up the blood flow into the microscopic glomeruli. There are 2.5 million nephrons in your kidneys, therefore there are 2.5 million of these afferent arterioles, 1 per nephron supplying blood into that ball of leaky capillaries. What kind of capillaries do we have in the glomerulus? Fenestrated, right? Very good. And remember that this is a portal system. The kidney is a portal system, and we know that that means that there are 2 capillaries involved. So the first capillary is the glomerulus, right? There's blood going into the glomerulus, but remember that blood goes into the glomerulus and comes out still red. So that blood is going into the glomerulus only for the purpose of being filtered, and there's no gas exchange here. Then the blood will leave out the efferent arteriole, travel to a second capillary bed, either the peritubular capillaries or the vasa recta, and there do the normal gas exchange. So here's the peritubular capillaries or the vasa recta, and then once we've done our gas exchange, then the blood will come back out, same backward story, interlobular veins, arcuate veins, interlobar veins, finally back in the renal vein, and the 2 renal veins left and right would then empty into the inferior vena cava and come back to the heart and start the whole cycle all over again. So this is coming from your book, just a quick review of the blood flow. So again, we're coming in the renal artery, branches into segmental, then goes down up along the columns. That's going to be the what? The columns are going to be the interlobar, between the lobes, if you will, between the pyramids. Then there's going to be the arcuate that go up into the cortex and arch around the interlobular, and then finally into those afferent arterioles. I just realized I was going backwards. So there's segmental, interlobar, arcuate, interlobular, then the afferent arterioles. So take a look at that overall blood flow. Once we get in here, and I'll show you the same exact slide in lab this week, we're going to see that there are 2 sets of capillaries. We already know that there's the glomerulus, and that's just about filtering. The second capillary, there's actually 2 choices. If we are talking about a cortical nephron, and on the right here, this is a cortical nephron. It says it in very small print, I know. This is a cortical nephron. It's up in the cortex. A cortical nephron is going to have the second set of capillaries wrapped around all of the tubules. So those are the peritubular capillaries. If instead we're looking at a juxtamedullary nephron, there's going to be a much deeper loop of Henle, and here we'll have these capillaries, and that's the vasa recta. So there's either the peritubular capillaries or the vasa recta, and those are true continuous capillaries where gas exchange occurs, and you'll see things go from red to blue. Overall, you know that each kidney has over 1 million nephrons, and the parts of the nephron are as follows. What do we have? The parts of the nephron, we have the renal corpuscle. And what is the renal corpuscle? It's a coming together of the glomerulus and the Bowman's capsule. Within the capsule, inquiring minds want to know, right? The outer layer or the parietal layer of the capsule will be made up of simple squamous epithelium, right? Just, again, very thin layer. The visceral layer of the capsule would have those podocytes on it. And I'll show you those podocytes again in a moment. And then in between, there's a capsular space that separates these 2 layers. So let's think about blood flow into the corpuscle again. So afferent arterioles bringing blood in. What did I tell you? The wall of the capsule, right? The parietal layer of the capsule is what? Simple squamous. The visceral layer, what's laying on top of the glomerulus? The podocytes, right? So the podocytes are what's kind of laying on top of the glomerulus. Blood is coming into these leaky capillaries, And only those things which are small enough to fit through the fenestrations are doing what? Getting into the space and leaving through the, this arrow is going where? That's the beginning of the proximal convoluted tubule. So only the smallest of things can get through those fenestrations. So the ammonia, the urea, the uric acid, the creatine, all those nitrogenous waste products are easily small enough to get through those fenestrations. Whatever does not get filtered out will leave out the efferent arteriole. And where is that going? Blood leaving out of the glomerulus is next going where? Is going to another set of capillaries, right? It's either going to go to peritubular capillaries or to the vasa recta, depending upon what kind of nephron we're in. Once it goes through that second set of capillaries, then it comes out and eventually gets to the renal vein. The nephron has polarity. And I started mentioning this today in lab today. I think I got sidetracked. I didn't finish this idea. So I want everyone to hear this very carefully. If I go back, I'm going to go back a slide, and I look at this image. This side of the glomerulus, right, is receiving blood, isn't it? So this side is called the vascular pole. That side is called the vascular pole. The other side, directly across, where we have the beginning of the tubules, that's going to be called the urinary pole. So the vascular pole, where all the blood supply is coming in or out, and the urinary pole, where we're beginning with the filtrate to make the urine. So now let me put those in words. So we have the vascular pole. Again, this is the side of the renal corpuscle that has the afferent arteriole coming in and the efferent arteriole leaving. And then the urinary pole is where you see the proximal convoluted tubule start. All of this tubing of the nephron can be divided up into some regions of all this tubing. And there is the proximal convoluted tubule, the nephron loop, also called the loop of Henle, and the distal convoluted tubule. These are all parts of a nephron. Right? So a nephron, you could say, is what? It's the renal corpuscle plus the proximal convoluted tubule plus the loop of Henle plus the distal convoluted tubule. Those four parts make up the nephron. Then many nephrons will drop into more ducting or more tubing, referred to as the collecting duct. And that's receiving the urine not from just one nephron, but from many nephrons. We're going to be going through what's going on physiologically here in a moment, but recall that the proximal convoluted tubule is largely involved with what? What's the major process happening in the PCT? Absorption or reabsorption. So this is where the kidney is doing what? Reabsorbing the majority of the water, reabsorbing the majority of the useful solutes. So wherever there's going to be a lot of reabsorption, what modification would you expect to see on these cells? Microvilli, right? We're going to increase the surface area substantially by having microvilli so that we can suck up all those things, right? We can reabsorb all those nutrients. Lining the PCT, simple cuboidal. Simple cuboidal. Then we get down into the nephron loop. It's this long U-shaped portion. There'll be simple cuboidal epithelium down here as well. There will also be some simple squamous. We'll talk more about this in a few minutes. But part of the nephron loop will be made up of what's called the thick segment. Part of it's made up of the thin segment. Makes sense. The thicker segment has cuboidal cells. The thinner segment has squamous cells. It's no harder. Don't make it harder than that. And we'll talk about what's going on there as we go up and down the nephron loop in a few minutes. And then there's the distal convoluted tubule. By now, we have reabsorbed most everything, so there's very few, if any, microvilli. Right? We've done our reabsorption by the time we get here, so we don't need the microvilli when we get to the DCT. The collecting duct is the last portion, not of the nephron, but of the tubing. And we'll see that the multiple nephrons will dump their fluid now into the collecting ducts. The collecting ducts will all go down toward the tip. What's the other word for the tip of the pyramid? They're all going to go down toward the apex, right? The point of the pyramid. And they're going to come to a papillary duct. We know what the word papillary or papillae means, right? Nipple-shaped. So at the apex, at the point of the pyramid, you will see what's called the papillary duct, basically a nipple-shaped or a drip spout where the urine is now being dripped into, and where's it going next? Once it leaves out of the papillary duct, it's going to drip into what? The minor calyx, right? So here is your flow. There is the more detailed flow, if you will. So you want to know that flow well. So can you picture it? If you are a nitrogenous waste molecule, if you're a uric acid molecule and you're traveling through the blood, and the blood now goes in the renal artery and is heading toward the glomerulus, you as a little nitrogenous waste molecule would find yourself where? In the glomerular capsule, the space, the Bowman's capsule, heading now down the proximal convoluted tubule, through the nephron loop, through the distal convoluted tubule. You'd be dumped into the collecting duct, through the papillary duct, into the minor calyx, the major calyx, the renal pelvis, the ureter, down to the bladder, and out the urethra. Right? So that would be your path as far as filtrate or waste products from the kidney through the anatomy of the urinary system. And that's what's shown here, right? So that's exactly what's shown here, going through a simplified drawing of this whole thing. Remember, these nephrons are microscopic. You will see them under the microscope this week in lab, and you will be expected to recognize what this looks like when you see it again. So I've alluded to the fact that there are two different kinds of nephrons. They have two different purposes, if you will. Cortical nephrons. The nephrons are where? More in the cortex. They're toward the surface of the kidney. So this one on the right is a cortical nephron. You see all of the tubing high up in the cortex. The loop of Henle is not very impressive at all and doesn't dip very far into the medulla. So this would be, let me draw a line here. Everything below this line is the medulla. Everything above this line is the cortex. Right, so we're definitely mostly up in the cortex with very, very little loop of Henle going down into the medulla. That's a cortical nephron, and this is going to represent about 85% of all the nephrons in your body. They have very short nephron loops, and what do they have? They have the peritubular capillaries. Versus this guy over here, this is a juxtamedullary nephron. They're more rare. There's only 15% of your nephrons make up this type. They have very, very long nephron loops. They're dipping way down deep into the medulla. And around them, one finds the vasa recta. Right, so vasa recta go with juxtamedullary. Peritubular capillaries go with cortical nephrons. We'll talk about what the difference is and why we need those two different types as we go through this. And you also shouldn't be surprised that the kidney has a series of nerves going there. And so, as is any other organ, if it was going to the heart, we call it the cardiac plexus. Since this is going to the kidneys, we'll say that this is the renal plexus. And these nerves are coming down and wrapping around the renal arteries. And we're going to see, and you already know this, that the sympathetic and the parasympathetic nervous system certainly have an effect on the kidneys. You know that when the bear is scaring you, that more blood is being shunted to your muscles than to your urinary system. And in a rest and digest time, you have more blood flow going to your kidneys. Not that your kidneys ever shut down. Not that your kidneys ever have the opportunity to completely shut down their blood flow. I want to make sure we get that. I think there's a simplistic idea that maybe I entertain or actually create. When I see the bear walks in the room and you're in a sympathetic surge and I say, hey, more blood's going to your muscles and less is going to your urinary system, I don't want you to ever think that it was shut off, right? It's simply reduced. And so this is a rheostat switch, more or less. It's never an on or off switch. It's simply a control of the amount of blood that can go to different regions. Your kidneys must continuously filter your blood to get rid of these waste products. Otherwise, in very short time, we become toxic. A person who's on dialysis needs to have dialysis every couple of days. Otherwise, they become very toxic, and these waste products begin to accumulate to a level that can become dangerous. So we don't have a chance to shut our kidneys off. So this renal plexus would be sending nerve signals to the kidneys, wouldn't they? Autonomic signals to increase or to reduce the blood flow into the kidneys. I think I just said this, but there's a couple extra things here. So you definitely have sympathetic signals going down to the kidneys, and when you have a sympathetic surge, it is going to stimulate the reduction of the glomerular blood flow. I kind of hate it when we say something goes up that goes down. So let's reword this. It stimulates a reduction in the GFR. So what does that mean? It reduces the glomerular blood flow. Now what does that mean? Less blood will be available to go into the glomeruli. Are you still filtering? Yes, but not as much. So your rate of urine production is going to go down. If you're stressed out, you're not urinating as often. Think about it, right? If you're ever in a stressful situation, you're not making as many restroom visits. Your kidneys are not getting as much blood. You're not filtering as much. You're not making as many waste products. Now, another thing, though, is that when you have this sympathetic surge or innervation, you're going to start secreting renin. And renin is an enzyme that is going to activate to restore your blood pressure. So we'll talk more about this in a minute, but your kidneys are definitely involved with regulating your blood pressure in a couple of different mechanisms. Finally, if you are in a parasympathetic moment, then this is coming into your kidneys through that good old friend, the vagus nerve. Remember the wandering nerve that travels all the way down to your organs, including your kidney? And that's going to increase your rate of urine production. So you just had that big meal, your body's digesting it, breaking down products, and your urinary system is also busier, right, filtering out your blood and getting rid of those products. Quick review. Let's start here, going a little bit more slowly, and make sure we've got this step by step as we go through this. Questions so far? Are you finding that reasonably reviewable, if that's the right word, right? Mostly stuff you've heard before, a couple new facts. But for the most part, things that hopefully you can dust off and recall quite easily. So what are the processes that the kidneys are doing? I think we know this, right? If we're making urine, what steps are involved in making the urine? So the very first step, we know that the kidneys are filtering. Right, so they're filtering our blood, and in this simplistic view, in this simplistic cartoon, what would this vessel right here be? Coming into the glomerulus, that would be the afferent arteriole. We're going to bring that blood into the leaky capillaries, and it's going to leave out the efferent arteriole. We got that. And then if this is a cortical nephron, where would that blood go next? To the peritubular capillaries. So the very first thing we're going to do in producing urine is filter. All right, we're going to take the blood and we're going to filter out the small particles. So really what we're filtering is the plasma, isn't it? To be more specific. And the things in your plasma are largely small. We're not talking about blood cells here, but everything else is pretty small. So basically the first filtrate is very, very similar to the plasma. Really is very, very similar as far as content of nutrients and things like that. Then this is, again, not showing all the tubule structures, but the next thing that's going to happen is that your kidneys are going to reabsorb. That's mostly in the proximal convoluted tubule, right? And what's going to happen here is that you're going to replace, or you're going to reclaim all of the useful molecules. So the water, the solutes, many the molecules that the body can use will be reclaimed through this process of reabsorption. Mostly in the PCT, also some of this going on in the loop of Henle. Then the third one is secretion. Now this word always gets me. I always have to think about this because when I think about secretion, I think about tears, right? Or maybe that's excretion. I just don't think about what this word means. Secretion is the opposite of reabsorption. So look at this arrow up here. So as we come out of the glomerulus, we're filtering, right? That's what that first arrow is. Reabsorption, what are we doing? We're taking things from the tubes and putting them back into the blood, aren't we? So that little arrow right there, that orange arrow going from the tube back into the blood, that represents the movement of molecules which are reabsorbed. Secretion is the opposite. So what is secretion? Taking molecules from the blood and putting them directly back into the tube. Now this happens largely in the distal convoluted tubule. And then finally, there will be water conservation. We'll see that the kidneys are going to be one of the primary places that are going to make sure you don't become dehydrated. Right? Clearly, if you're well hydrated, you're going to urinate a lot out. But if you're dehydrated, the kidneys are going to work with your hormonal signals to retain fluid. And the thing it doesn't have on here that I want to maybe act as a 5 is as you release urine, that word is what? Excretion, right? So your excretion is the release of the urine. Really not the urinary system, it's done with it, right? The urinary system's done. It's done with it, and now what's left over is going to be released from the bladder and through the urethra. So I'm going to be breaking this down, urine formation. Remember that what is first filtered is just that. It is complete plasma. There's a lot of good stuff in there as well. So the first thing we're going to do is convert plasma to urine in 3 stages. Let's start off with the first step. The first step is going to be this filtration step, right? The glomerular filtration. And we know that's only happening, as it says, in the glomerulus. First step. Then we'll go on and talk about reabsorption and secretion, and then finally we'll talk about water conservation. So whatever gets out of the glomerulus is going to collect where again? Whatever leaks through those fenestrations is going to collect in the Bowman space or the capsular space. This is not urine by any means. We're going to call this simply filtrate, right? It has been through the filter. We're going to call this solution or this stuff filtrate. As it continues through the tubes, it's going to become urine, and it will be added to and taken away from through the processes in the nephron. So we're going to call this tubular fluid at this point, and it's going to start a journey down the proximal convoluted tubule, and it's going to go down the loop of Henle and finally come up at the distal convoluted tubule. Along the way, we're going to remove stuff and add stuff. By the time we get down to the collecting duct, then we have urine. Okay. The only thing we can modify at the collecting duct is water. So when we get down to the collecting duct, we can suck more water out of the urine to retain fluid, but as far as moving molecules in or out like sodium or potassium or calcium, we're done. So by the time the filtrate gets to the collecting duct, that is urine. The only modification that can still be done in the collecting ducts will be water conservation. So here's just a beautiful little picture, right? Beautiful. What are we looking at? In this color-enhanced micrograph, this big old cell on the top is a podocyte, right? The podocyte is that cell that is reaching around, and we know that pod means what? Pod? I'm going to go with the podiatrist because I'm having foot issues, right? So podocyte is a cell with foot-like branches or extensions. So these podocytes, and we see these long foot-like or arm-like extensions coming out from the cell. Now, what those podocytes are going to, or what those podocytes are doing is they're creating a second level of filtration. Remember that molecules have to get out of the blood and into becoming filtrate. They have to get through the fenestrations and slide through these little filtration slits, which are formed by the podocytes. These podocytes are also giving the capillaries some structure. Remember what do we know about capillaries? They are they're not very impressive as far as structure, are they? Right? They're very weak. They're only made up of a simple squamous epithelium. And now I'm telling you that these capillaries are fenestrated, so they have holes in them, so they're even less structurally intact. And what I've not told you before is that the renal capillaries get pretty high pressure. right? When we're talking about capillaries getting low pressure, that's true of most of the capillaries in the body. But in order to do the job of filtering, the kidney is receiving blood under pretty good pressure. And so these renal glomerular capillaries are under more pressure than most capillaries to get the filtration job done. So we need some extra support, and the podocytes are going to assist in that. So I need you to take a little magic school bus ride with me down to the microscopic level of the glomerulus. And what we have here, this is the glomerulus. So we're in the blood capillary. Here's a red blood cell, right? So we're in the kidney, we're in the glomerulus, and this is the stuff that is about to be filtered. What's it going to filter through? The fenestrations, because this is the simple squamous epithelium. This is the simple squamous epithelium, as is this. But there are those fenestrations leaving or available in the wall of the capillary. Then, just a short distance over, this big yellow thing represents the podocyte. And the podocyte, in those little foot-like extensions, also create filtration slits. So between these little extensions are additional slits. Anything that's going to leave from the plasma, go through the fenestrations and get through those filtration slits must be less than 8 nanometers in diameter. Okay, what's a nanometer? 10 to the minus 9. We're talking 1/1000th of a meter. We're talking small, right? So what kinds of things can easily get through? You have to be as small as a molecule of water. Electrolytes like what? Sodium, potassium, calcium. Glucose is small enough. Amino acids are small enough. Individual fatty acids, vitamins, and then that's all the good stuff, right? And oh, by the way, also sliding through those slits are the waste products, urea, uric acid, and creatinine. Okay, so in order to filter through, we had to let go of some very important molecules that the body is going to have to now reabsorb. What cannot get through these filtration slits? In a normal kidney, you shouldn't be seeing white cells or red cells, right? Those are too big to get through the slits. Look how big that red blood cell is, right? There's no way that red blood cell is going to fit through that little hole. There's also going to be plasma proteins, things like albumins and your immunoglobulins. Those should be too big to get through. Some do occasionally get through, right? A little bit of protein in your kidney, not a major issue, not a major concern. Large anions. What are anions? Things that are what charge? Negative. Okay? So large anions, and most of your hormones are not going to be able to get through, right? They're just too big. So we've got most of the stuff is being kept behind. Only small things are able to get through this double filter, right? We've got the fenestrations and the filtration slits, a double, almost like a double piece of filtration paper or filter paper. Very, very small molecules can get through. So the process of glomerular filtration is simply the exchange of small molecules getting out of the blood plasma, passing through the capillaries of the glomerulus into the capsular space. And to do that, they had to also pass through the filtration slits of the podocytes. So I've already said this, but let's just put it into words and add a couple details. So there's actually 3 barriers here. I only described 2 of them. The first barrier, the first opening, would be the fenestrations of the endothelium of the capillaries. Again, really small molecules can get through this. Blood cells can't get through here. Highly permeable, like lots of movement of molecules. Then there's also a basement membrane here. Basically a little bit of a layer. And this basement membrane actually helps to exclude molecules. What did I say? Things had to be how small? About 8 or so nanometers, right? How big are these slits? 70 to 90. Okay, so whoa, wait a second. You got big fenestrations, but I'm telling you only really small things can get through. And the reason is the basement membrane has, as a part of its nature, a highly negative charge. That means that it's going to repel other negatively charged molecules. Most of the proteins in your body, even if they could get through the hole, are also negatively charged. So it's like two negative magnets coming together, right? They repel each other. So if a protein tried to get through the big opening, it would be repelled by the negative charge of the basement membrane. So just to give you an idea of how effective this is, your blood plasma is about 7% protein. How much protein can actually get into your filtrate? 0.03%. Not to memorize the number, but to realize this is a very effective barrier. Only a very, very small amount of protein will get into your urine. That's why if there's a trace amount on your chem strip in lab this week, you're like, okay, a little bit of protein, okay. Right? A little tiny bit is okay. A trace, that's fine. Rather quite normal. And then thirdly, so you've got the fenestrations, you've got the basement membrane with that negative charge, and then finally you've got the filtration slits, like we mentioned, which are the podocytes. The pedicels are the structures. So the podocytes have these big arm-like structures called pedicels that wrap around the capillaries. And again, they also create a negative charge. So you've got a lot of things going on here. You've got both size and charge helping to determine what can or cannot get through these slits. In the end, here's kind of the rule. Almost any molecule smaller than 3 nanometers can freely get through. And we've already listed those things on the other slide. So anything that's really, really small can get through these 3 filters, if you will. Some smaller things cannot get through, even though they're small, again, or because they're bound up. Remember, you may have heard me say that iron doesn't float around in your blood by itself. It's usually bound to something else. So that means that iron usually is not going to show up in your urine because iron is already bound up to a larger protein, so it can't fit through the slits. Thyroid hormone, remember that was one of those hydrophobic proteins. Even though it was very, very small, it was always being carried by a carrier protein. And so thyroid hormone, even though it's very small, cannot easily end up in your urine because it's already bound to a larger protein in your blood. If it was unbound, right, if it was free, it would be able to pass. So unbound iron and unbound thyroid hormone could, because it's so small, get through, but usually it's bound up. Now, when will you start seeing the wrong stuff in your urine? What would cause you to start seeing blood cells or start seeing larger proteins in your urine? Well, something's going on with your glomerulus, right? Your filtration slits, the overall filtration membrane must be compromised if you are beginning to see large amounts of protein, if you're beginning to see blood cells in the urine. It makes sense, doesn't it? So what would cause that to happen? Well, let's go back up. Proteinuria, we'll talk about this in lab a little bit this week. Proteinuria is just a generic term, meaning that in the urine, not emia, but urea. So in the urine, what is there? A high amount of protein. What is the most abundant protein in your urine? Albumin. So they're sort of the same thing. If you're going to have a high amount of protein in your urine, likely it's going to all be albumin, because albumin is not only the most abundant, but the smallest protein, the one most likely to show up in your urine. And so you might also be said to have albuminuria, right? Same thing, proteinuria or albuminuria. This is going to be a protein of presence of protein in your urine. You could also have hematuria, which would be the presence of blood in your urine. So again, what would cause those molecules to show up, or those things to show up in your urine? Some damage to kidney disease of some sort. It could be an acute infection of your kidneys. It could be a chronic problem with your kidneys. There could be trauma to your kidneys. You've been bumped or hit, and your kidneys are dealing with that damage or that inflammation. Anything like that could cause you to start seeing in your urine for a short while, or longer chronic problems, see these molecules as an indication of kidney damage. even distance runners and swimmers can experience temporary proteinuria or hematuria, again, because what are they doing? They're in this constant strenuous activity. The blood is going where? Largely to the muscles during this. That's going to reduce the perfusion. What's that word mean? Perfusion is the amount of blood going to the kidneys, and it turns out the kidneys get really pissed off when they're not getting enough blood. So if they're not getting enough blood, they start revolting, and the glomerulus begins to deteriorate. Well, if the glomerulus deteriorates, guess what's happening? Things are going to get through the slits that wouldn't normally get through. So a person who's doing strenuous exercise for long term, again, distance swimmers, distance runners, might experience at the end of that exercise, if they check their urine, they might see proteinuria or hematuria. It's a temporary thing, right? Because the glomeruli are screaming at you, because not only were the kidneys not getting enough oxygen, sorry, they weren't getting enough blood, they also weren't getting enough oxygen. So when I say the kidneys get upset when they don't get blood, that's true, but really what's driving their frustration, let's give the kidneys a personality, but what's driving the kidneys to get upset isn't so much the low blood as it is the low oxygen, right? So they need oxygen. And so the kidneys become hypoxic, the glomeruli begin to break down, deteriorate, they become more leaky than they would normally be, and we start seeing blood and urine in the urine. Blood and protein in the urine. It's good to see urine in the urine. Okay. Ty.
To what extent is that temporary? Like, is that
I don't, good, the question is how temporary is that? I mean, I don't know how long you would see it following the exercise. If it's a day or hours, I really don't know. So good question. I should look that up.
Distance runners usually running every day, most days of the week.
But again, your body gets used to things too, right? So if your kidneys have become accustomed to your running often, they may not be so upset. But maybe the time you go longer than normal, or the time you go beyond, you're in competition, you're pushing yourself beyond, then your kidneys would be more frustrated or unexpecting of that hypoxia. So I would think your kidneys would adjust to this as well. Okay. So that's the filtration side of things. Does that make sense? So far everything's okay there? So I've already told you that the capillaries going to the glomeruli are getting a higher blood pressure than any other capillaries in your body. Most capillaries in your body are going to get somewhere around 10 to 15 millimeters of mercury. Your glomerular capillaries, 60. So they're getting a lot more blood pressure. So they're receiving, I mean, and if you look at it, you will appreciate that the renal artery is a pretty big artery, right? It's a pretty big branch coming off. It's not that far from the aorta or from the aortic arch. And so there's still a lot of pressure going into the renal arteries and going into the afferent arterioles. When you're looking at the model, remember that the afferent arteriole is always going to be bigger or larger than the efferent arteriole. That is, there's more blood going into the glomerulus than what is coming out. Okay, so you know that fluids only will go down a tube if there's a pressure difference. So we're going to talk about that pressure difference here just a little bit. And the hydrostatic pressure would be the pressure established by what? Hydro, water, fluid, right? So the fluid pressure on the glomerular space, there's going to be about 18 millimeters of mercury. And this is going to be because you have that high filtration rate and you've always got fluid accumulating in the capsule. I'll show this to you in a picture in a moment. Then there's colloid osmotic pressure. Where have we heard that word colloid before? Colloid, you've heard these words before when we were dealing in regular capillaries. Remember in capillaries I said there was hydrostatic pressure, that was the amount of pressure the blood was being pushed into the capillary bed on the arterial side. And then there was always reabsorption of fluid at the venous side because there were colloid or protein sucking the water back into the blood. It's the same story here. The colloid osmotic pressure is about 32. And when we look at these two numbers, what you're going to see is that there's a net filtration pressure of about 10 millimeters of mercury outward. So let's look at the picture. It's a lot easier to look at. So what this is telling me is that blood is coming in to the afferent arteriole and into the glomerulus under pretty good pressure, right? The blood hydrostatic pressure is 60 millimeters of mercury. But there is a colloid pressure and a capsular pressure pushing back, such that the fluid going down your proximal convoluted tubule is only under a pressure of 10 millimeters of mercury. Remember, there has to be a pressure difference, right? For that fluid to move down the tube, there has to be a pressure difference. It's only 10 millimeters of mercury. That's not a lot of pressure. Okay? So just know there's about 10, this is the key number, about 10 as you are going into the tubes of the kidney. Now again, this is why high blood pressure can be so devastating to the kidneys. If you have hypertension, not only is the rest of your body and all of your vessels seeing that higher pressure, making you more prone for aneurysms and issues with your vessels, but your kidneys are getting an unusually high percentage of this hypertensive pressure going in. So if your kidneys are receiving too much pressure, then what's going to happen? Over time, it's going to lead to rupturing of your glomerular capillaries. They're going to break down, and once they're broken, they don't restore, they don't rebuild. And that's going to start scarring up your kidney. That would be considered nephrosclerosis. That term makes sense? Nephro, kidney, sclero, hardening. So nephrosclerosis and atherosclerosis. So you will start getting restriction of blood going to the kidneys. I think I mentioned to you that one of the hot spots for atherosclerosis were your carotid arteries as well as the renal arteries, if you go back and look in the last chapter. So the blood flow going to the kidneys can become obstructed through atherosclerosis. Well, what's that going to do to the pressure? Right, if you block it, you're going to get less and less blood flow. And what am I telling you? If the kidneys aren't happening, if they're not happy, if they're not getting enough blood, if they're not getting enough oxygen, the glomeruli get pissed off. And they stop working, and now you're in kidney failure, right? So we want to protect our renal arteries by protecting our overall blood pressure. So one of the reasons we say that hypertension is a silent killer, right? You're walking around for 20, 30 years, and life is okay. But for those 20 or 30 years, you've been slowly destroying your kidney function. And I'll mention this again later, but kidneys are so redundant. What does that mean? There's so many nephrons that we all know we can live with one kidney. Right? You can live with one kidney, no problem. So you can live with 50% kidney function from the very beginning and do very, very well. In fact, you can live with a quarter of a kidney and still do fine. Right? You can live with a very small amount of renal function and everything as well. It's not until you're down to about, plus or minus, 10 to 20% of renal function that you might start noticing problems. You've already destroyed 80 to 90% of your kidney function before you are declared as having kidney problems. And once those 80 to 90% of your kidneys are wiped out, they don't restore. So once you are having kidney issues, you've only got 10 to 20% of your function left, and all we can do now is try to protect that last 10 or 20% and keep you alive, and maybe have to put you on dialysis if your kidneys really start to go below that, and then start looking for kidney transplants, because we can't do without our kidneys, right? We can't allow these toxic products to accumulate. Our blood goes acidic, and if our blood goes acidic, all things start breaking down. The pH of our blood, the enzymes, your brain, everything shuts down when we become acidic. Okay, so that's sort of the big global overarching reason why we check our blood pressure, and we want to make sure things are good. Questions so far on all things related to anatomy or flow of blood so far? Take a little break right here, and we'll move into the rest of this conversation for the afternoon. So what are we thinking? Anything so far? Structurally, blood flow, we getting the idea, right? Kidneys are pretty important. You got to have blood flow, got to have oxygen, and we're going to see what happens here with a couple of numbers. I know you guys all love numbers, but let's take a look at this. The GFR, the glomerular filtration rate. Don't make this into some complicated thing. What is it? the amount of filtrate formed per minute by both of your kidneys collectively. So your GFR. In the average male, you are making, guys, about 180 liters a day of filtrate. Ladies, about 150. That's huge, right? How much blood do you have? 5 liters or so. And out of those 5 liters, about half of which is plasma. So let's be generous. You have 3 liters of plasma, right? 3 liters of plasma in your body. And out of those 3 liters of plasma, every day you're making 180 or 150 liters of filtrate. What does that tell us? Tremendous amount of recycling going on, right? So you're constantly recycling and filtering and recycling over and over and over. Look at the rates. 125 mils of filtrate per minute. This is huge, right? This is a huge amount going on. And what is directing this has a lot to do with your pressure. So this NFP is your net filtration pressure. In other words, the more pressure you have coming into the glomerulus, the more filtrate you'll make. Yes, that makes sense? The more pressure coming in, the more filtrate that will be filtered, and the more filtrate that you would produce. The other thing is the K is a coefficient, pretty much a constant, that we're not going to play with. So really all I want you to see is that the greater the pressure, the greater the GFR, right? The greater the filtration pressure, the greater the GFR. So that means that you're producing 50 to 60 times the amount of blood in your body, complete, every day. Now, of that filtrate, you're only going to urinate 1 to 2 liters a day, right? We know somewhere around 1.2, 1.5 liters, maybe if you're well hydrated, a couple of liters. So that means that you are reabsorbing 99%, right? So you're making this stuff hundreds of liters a day, 150, 180 liters a day. You're going to reabsorb 99% of it. You're only going to end up with 1 to 2 liters of filtrate, pretty concentrated filtrate, with specifically the uric acid and the urea and the creatinine, right? You're going to be concentrating those nitrogenous waste products, and most of the good stuff, the glucose, the water, the sodium, the potassium, is going to be largely recycled. Now, what's going to happen? Let's think about this. If the GFR is too high, if the glomerular filtration rate is too high, then what's going to happen? You're pushing blood, or you're not blood, you're pushing fluid through the renal tubules too fast. Now, the renal tubules, that's a collective word for what? All the tubes of the nephron. Right? So you're pushing fluid through the PCT, through the loop of Henle, through the distal convoluted tubule too quickly. Well, if it's moving too fast, what can it do? It cannot reabsorb properly. So your urine output would rise, right? The amount of fluid that would get out of your kidneys into your bladder would increase, and you would have a greater chance of becoming dehydrated and depleting your electrolytes because you're not reabsorbing at the proper rate. What if instead your GFR is too low? Well, then you're going to be doing what? Reabsorbing not only what you need, but also reabsorbing some of the crap. Right? So you would start reabsorbing your waste products. It's sitting there too long. And you would start to accumulate, have azotemia, which again was the increase in the metabolic waste, the increase in the nitrogenous products. So we need to keep our GFR well regulated. So you're hearing that word homeostasis in your head, right? Oh, he's going to talk about homeostasis here, right? We have to keep this we have to keep the GFR within a narrow range of normal so that our kidneys can continue to function properly. Now, the GFR is going to be constantly monitored moment to moment by one of 3 ways. There's going to be an autoregulation, a sympathetic nervous control, and a hormonal control. Gee, we kind of talked about these 3 things already once before when we talked about capillaries, didn't we? Remember we talked about local control, We talked about hormonal control and neural control, how it is that blood flow is increased or decreased based upon nervous signals, hormonal signals, and the local pH changes. So we've already sort of heard this story, but now we're only dealing with the blood flow in the kidney, regulating the flow, the GFR. So let's go through these three ways. First, we'll start off with renal autoregulation. Again, what does this suggest? The kidney is doing this on its own. It's not involving the nervous system. It's not involving any hormonal signals. This is cool, right? This means that the nephron is able to adjust its own blood flow. Now, keep in mind, you have about 2.5 million nephrons. Each nephron has the ability of auto-regulating its own flow. But if you're a nephrologist, right, if you're a doctor looking at the overall function of your body, kidney nephrologists and doctors talk about the kidneys as if they are one big nephron. Right? The kidneys collectively, these 2.5 million nephrons are collectively doing what? We can't look at each individual nephron and figure out what's going on. So we talk collectively as a system. So we say that the whole kidney is basically one big nephron, right, doing the collective job of all of these nephrons. So number one, the kidney can auto-regulate its own GFR, and this is going to be, and what's cool is that the kidney can maintain a relatively stable GFR even with changes in your blood pressure. So as your blood pressure is changing, as you sit up, stand up, run, exercise, your kidney is able to auto-regulate its GFR and keep it pretty much within a set range. There's two ways that the kidneys are going to maintain this auto-regulation: myogenic feedback and tubular glomerular feedback. There's a mouthful. Myogenic tells me what? Autoregulation at the level of myo, muscle, genic, formed by or produced by the muscle. Tubular glomerular feedback, in a moment I'm going to describe to you this feedback, but it must be what? Something going on between the tubes and the glomerulus. There must be some sort of feedback going on between the proximal and distal tubes and the glomerulus. Let's start off with myogenic mechanism. Now, what do we know about smooth muscle? This goes back to exam 3. And we learned that smooth muscle, when it is stretched, does what? If I stretch smooth muscle, what does it do back? It pushes back, doesn't it? So if I stretch if I'm eating and I stretch the smooth muscle around my gut, what's my smooth muscle going to do? Contract and squeeze back. So there's this pushback. So this is the idea that if smooth muscle is going to contract when it is stretched. Can you think about where I'm going already? So that means that if I have increased blood flow into the glomerulus, and I have increased blood flow in the afferent arteriole. Remember, an arteriole, it's an arteriole. It has a lot of what in it? Pressure, but also muscle, right? The tunica media. We're still talking about an artery. And so there's a tunica media. We know that the tunica media is made up of smooth muscle. So if there's more blood traveling through the afferent arteriole would be stretched. The smooth muscle would be stretched. And what does smooth muscle do when it's stretched? Contracts or would constrict back. So that means if I've got too much blood flow going into my afferent arteriole, what's the arteriole going to do? Constrict back. And what is that going to do? Reduce the blood flow. Right? At the level of muscle, myogenic. If too much blood's coming in, that vessel is going to squeeze and restrict blood flow. What's this going to do? Reduce the blood flow going into the glomerulus, which would do what? Again, remember I said that increasing pressure increases GFR? So if I decrease the pressure, then I would be decreasing the GFR in that particular nephron. Right? So that means that the arteriole is going to constrict. Now, what happens when the blood pressure falls back down? When the blood pressure returns back to normal, then what would the afferent arteriole do? Relax, right? We no longer constrict, it would go back to being relaxed, and that would do what? Allow more blood into the glomerulus, and this constant myogenic squeezing and relaxation of the afferent arterioles is going to do what? Pretty much keep our filtration levels stable. This is myogenic autoregulatory mechanism to keep your blood flow and your filtration rate as stable as possible. That's one way. The second way is also autoregulation. This is tubuloglomerular feedback. I'm going to have to introduce a little bit more anatomy here for you in the kidney. That is brand new for you. So this is a mechanism whereby the glomerulus, right? The glomerulus is going to receive feedback information from the tubes itself. I need to introduce this thing called the juxtaglomerular apparatus. You'll see it abbreviated the JGA. And what this is a structure, a series of cells that are at the very end of the nephron loop. Okay, so where am I? If I'm at the very end of the nephron loop, I'm about to go into the what? I'm about to go into the distal convoluted tubule, right? If I'm at the distal end of the nephron loop, at the end of the nephron loop, then I'm about to head in, into the distal convoluted tubule. And what we're going to see that maybe never picked up before is that the nephron loop comes back up and comes in direct contact with both the afferent and the efferent arteriole at the vascular pole. Let me say it again, then I'll show you a picture. The nephron loop comes back up out of the medulla, comes back up, and is going to go directly between the afferent arteriole and the efferent arteriole at the vascular pole. I'm going to jump ahead and show you a picture of this and then come back. So I jumped ahead a couple of slides. What did I call this end of the glomerulus? Vascular pole. And this end was the urinary pole. Vascular pole because I have the afferent arteriole coming in, the efferent arteriole going out, all the blood supplies coming in here at the vascular pole. On this end, the urinary pole is where the filtrate begins its journey down the proximal convoluted tubule. Okay. This, it's on the models upstairs. I just never introduced it before. So there's the afferent arteriole coming in, the efferent arteriole coming out, and there's this, I don't know, looks like some sort of tube coming up right in between them. That tube is the nephron loop. Okay, so the nephron loop, right, the filtrate went out the proximal convoluted tubule, went all around its crazy little convoluted tubule, went down the nephron loop and comes back up the nephron loop right here, and is in direct connection right next to both the incoming and outgoing blood flow. That total area right there is this whole area, this box, if you will, right here, is the juxtaglomerular apparatus, the JGA. Already, think ahead, what am I telling you? That the flow of filtrate through this tube can do what? Feedback and regulate the amount of blood coming into or out of the glomerulus. So I want you to see the picture of it. Let me go back now and talk you through a couple slides, and we'll come back and look at that picture in a moment. So again, going back to the beginning of this tubuloglomerular feedback, there is a mechanism in place such that the glomerulus, right, the capillary, can receive feedback status information from the downstream nephron loop. Okay, and again, this happens at the JGA, at the juxtaglomerular apparatus. Now, there's three kinds of cells here in the JGA. Okay, in this area, in that little box that I drew in, there are three special kinds of cells that make up this JGA, this juxtaglomerular apparatus. Number one, the macula densa. Now in lab this week, one of the slides has the macula densa circled, and I'll point that out to you in lab. It won't make sense to you, well, now it'll make sense when you see it again after hearing this. But the macula densa is going to be a patch. And what does that word macule mean? Macule means stain or a spot, right? We learned that last semester. So it's a spot, a little patch of cells that are closely spaced together inside the nephron loop on the same side that are facing the afferent and efferent arteriole. And these macula densa cells are going to be sensing changes in the flow, how much is flowing through the nephron loop, as well as the composition of the filtrate, and will, if necessary, secrete a paracrine. Now, what's a paracrine? It just says a paracrine here. But what is a paracrine? Not an endocrine. An endocrine would be a hormone that travels far, far away. A paracrine would be a substance released by one cell that influences a neighboring cell, a close-by cell. Okay? So we're going to see that the macula densa cells can release a substance which will act on the neighboring JG cells. That's the second kind of special cell here, the juxtaglomerular cells, the JG cells. What are they? Smooth muscle cells. So the JG cells really are specialized smooth muscle cells, and we'll see that they can get stimulated by the macular cells, and they can cause the arterioles to either dilate or to constrict. Also of significance, the JG cells are the cells that contain and release renin. So they release renin, and they release renin whenever there's not enough blood pressure. So whenever there's a drop in blood pressure, then these JG cells are going to release renin, and we'll talk about what that renin's doing more completely maybe today. Finally, there are meningeal cells. Now, break that down. Mesangial. Angio. What does mes mean? MES? Mesoderm, the layer in the middle. Mes means middle. And what does angio mean? Vessel. So where are the mesangial cells? In between the vessels. What vessels? The afferent and the efferent arterioles. Okay, so if we don't memorize these words, but we break them down and think about what they mean, they'll speak to us, right? Mesangial. Okay, it's speaking to me. What? Cells that are what? Between or in the middle of the vessels. Okay, mesangial cells. So they're right in between the afferent and efferent arterioles. And we're going to see that they're in direct connection with the macula densa cells and with the JG cells. And they're going to work. They're going to talk to each other. Okay, so this talking back and forth is this whole JG glomerular apparatus feedback that I'm talking about. So let's take a look at the anatomy here and what's going on. In fact, I think I almost want to go back quite a few slides here because I want you to see the big picture of something. I'm going all the way back toward the beginning of our conversation, right here. Because I want you to see this overall structure of the nephron that maybe never was obvious to you before. So as I look at this, let's follow the tube. And I know this is small, but let's do this. Blood's coming into the glomerulus through what? Afferent arteriole. Goes into the glomerulus, filters out. That filtrate's going to start a journey down the proximal tubule, right? Windy, 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 windy. Down the nephron loop. Back up. And look where it goes. Boop. Where's that nephron loop run right into? Right in between the afferent and the efferent arteriole. Look over here at this other nephron. Same idea. As the nephron loop comes back up, boom, boom, it runs right into between that afferent and efferent arteriole. You never noticed it before. It wasn't critical that you noticed it. But now you see that as that nephron loop comes back up, it's speaking back to the glomerulus, isn't it? That's this juxtaglomerular feedback, right? So let's go back forward to where we were, now that you've seen the big idea here. And go back down to this picture and imagine where we are. So the little inset at the top left reminds us, right, that this nephron loop coming up, right, this nephron loop coming up was down in the medulla, right, came back up and is running, boom, right in between the afferent and efferent arteriole. Okay, so again, this box is the JGA, the juxtaglomerular apparatus. Within this area, what do we see? The lining of cells, this group of cells inside the nephron loop, facing the arterioles, that cluster or spot of cells, that's the macula densa. The little smooth muscle cells that are wrapped around up here, those are the JG cells. What did they do? They were muscle cells that what? Produced renin and could speak with the other cells through paracrine molecules. And then I told you there were meningeal cells. And let's look at where they are. It's pointing in two places. It's saying the meningeal cells are right here. Right? Mesoangeal, between the vessels. They're right in the crux between the afferent and efferent arterioles. But also, there are some meningeal cells over in here, these purple guys. So again, where are they? Between the vessels, in the middle of the vessels. But when I talk about meningeal cells, I'm really going to be referring to the ones right here, right stuck in the middle of those three. So over on the left, or sorry, on the right, it tells us what's going on here. Again, this is autoregulation, myogenic and juxtaglomerular working together to regulate flow of blood and filtrate production in the glomerulus. So if the GFR rises, GFR, glomerular filtration rate, if the rate of fluid going through the glomerulus and making filtrate gets too high, what's going to happen? There'll be more fluid in the tubes, wouldn't there? That increased fluid is going to stimulate the macula densa, right? These cells right here, right? They're going to be saying, hey, the pressure in this tube there's a lot more fluid in here than what I'm accustomed to, and it's going to signal the macula densa cells, and they're going to secrete their substance that are going to stimulate these JG cells. The JG cells are now going to squeeze down. Where are they? What are they wrapped around? Which arteriole? They're wrapped around the afferent arteriole largely, aren't they? So if those guys, if those JG cells, which are smooth muscle cells, if they squeeze down, what's going to happen? I'm going to reduce the amount of blood flow coming in and therefore return the GFR back to normal, aren't I? Okay. And those mesangial cells also can squeeze, and they will also constrict the capillaries and reduce the amount of blood flow going through. Now turn it around. What if the GFR begins to fall? Just flip it. Now the macula would relax, right? There would not be any stimulation of the macula, and that would then tell the juxtaglomerular cells and the mesangial cells to do what? Relax and restore blood flow back to normal, and hopefully that would increase the GFR back to normal. So we've got two things going on, myogenic and this juxtaglomerular feedback, both of which are non-hormone, non-nervous system signals, and this is pretty much what the body's doing, right? The body is going to maintain your GFR in a pretty tight, so the GFR fluctuates very little, right? Your GFR stays rather steady. However, your autoregulation mechanisms cannot compensate for extreme changes. So within a normal blood pressure range your kidneys will keep their GFR nice and flat, nice and steady. Homeostasis working at its best. However, if you have, I should go back, if your mean arterial pressure is between 90 and 100 millimeters of mercury, then the GFR will remain stable. What's the MAP? Do you remember that? Remember what the MAP? If the MAP starts to go quite low, then if your MAP goes low, then the glomerular filtration and the urine output will go down. So you wouldn't want that to happen, would you? You never want your filtration to go down or urine output to cease, because then you're not getting rid of waste products, are you? Now, another thing to think about, what if you had hypovolemic shock? What is that? What is hypovolemic shock? A drop of blood volume. Again, right? If you have a drop in blood volume, then your kidneys aren't going to get enough volume and you would have some kidney issues. So when the body cannot maintain through autoregulation, then we're going to see hormones kick in and the nervous system kick in. But let's take a look at this overall idea. So negative feedback, let's start here. If you had a high GFR, a high glomerular filtration rate, what does that mean? You've got too much fluid, too much blood pressure, and too much fluid going into your glomerulus, and you have too much fluid being produced. That's going to give you too much flow of fluid through your renal tubules. That increased flow is going to be sensed by the macula densa, that group of cells in the rising part of the nephron loop. Those are going to release paracrines, which are then going to cause the constriction of the afferent arteriole. What's going to squeeze that? What cells are going to squeeze that afferent arteriole? Smooth muscle cells, specialized smooth muscle cells called the JG cells. Right? The JG cells are a type of smooth muscle cell. And then because they're squeezing down on the afferent arteriole, that's going to ultimately reduce the GFR. But what if that's not enough, right? What if the kidneys are not able to maintain flow within the autoregulation? Then, or also, we can have control through the sympathetic nervous system. And you know, as we've mentioned, that there's definitely nerves going to the kidney as part of that renal plexus. And what happens is that good old epinephrine, right? Good old epinephrine is going to constrict the afferent arteriole. Okay, so what's epinephrine doing? Norepinephrine and epinephrine are both released by the adrenal gland when you're scared, right? When you're in times of stress. And we know that that's going to increase your fight or flight responses, so more blood flow would go to your muscles. Well, how does the body allow for more blood flow to go to the muscles? Because the kidneys are going to get less blood flow. And so the epinephrine is going to constrict the afferent arterioles. Okay, fine, no problem, right? And this is going to allow more blood to be available elsewhere. Now, during a sympathetic time, you would have reduced GFR, less urine output, because your body is redirecting that blood to other places. It'll go low. It'll never shut off, but it will get a lot lower. Right? The GFR will definitely go a lot lower. That's the sympathetic. Not much to know, right? We already know this. We know that during sympathetic surges that the kidneys are receiving less blood. Now, the last portion is hormonal. And this is a really important slide. So put a big old star by this one. I know this is small. This is figure 2315 in your textbook. I know you're going to see this in your Connect assignments as well. There is a enzyme hormone system, and you'll see it referred to as the RAA system. RAA. And RAA stand for renin, angiotensin, aldosterone. RAA. So when the autoregulation isn't enough, and when the sympathetic nervous system doesn't make the necessary changes, now we have some hormones that can come to the rescue. Renin is what's going to start this whole process. Renin is made by what cells again? made by those JG cells, and they make renin or release renin when blood pressure is too low. Okay? So the blood pressure to the kidney overall is too low. We know the kidney doesn't like that. So the kidney takes it upon itself and releases renin. Now what renin is, it's actually an enzyme. And what that enzyme is going to do is cleave or convert a molecule called angiotensinogen into angiotensin 1. Look at the ending of those words. It's very similar to fibrinogen becoming fibrin. Right? Remember when blood clotting cascades happen, the fibrinogen, which is floating in your blood, is converted to fibrin. Here, this enzyme renin is going to convert a molecule called angiotensinogen and is going to convert it into an active molecule called angiotensin 1. Okay, so now you've got angiotensin 1 traveling through your blood. Right? Angiotensinogen was already in your blood. It was already there. It's just already in your blood. When renin is released, it converts it to angiotensin 1. That angiotensin 1 is now circulating through your body, in your blood, and this is kind of odd, but in your lungs, your lung tissue makes an enzyme, as do your kidneys, make an enzyme called ACE, angiotensin converting enzyme, ACE. And angiotensin converting enzyme, ACE, converts angiotensin 1 into angiotensin 2. Now that is now the active working hormone. And we're going to see what angiotensin 2 does in a moment. Have you heard of people going on ACE inhibitors? Right, for blood pressure control, ACE inhibitors? Well, we're actually monkeying around with this RAA pathway. When you give someone an ACE inhibitor, you are not allowing them to do what? You're interfering with their body's ability to make angiotensin 2. And we'll see in a moment what this does to blood pressure. Well, you already kind of know, right? What was this whole cascade about? To increase blood pressure, right? And so if you're giving someone an ACE inhibitor, what are you saying? Your blood pressure is already high enough, buddy. We don't want to increase it any higher, so we're going to give you an ACE inhibitor, which is going to help keep your blood pressure low. So let's go on and figure out what happens here. Now, I know this is small, but again, there's a drop in blood pressure. Your kidneys sense it, and they release an enzyme called renin into the blood. Renin is going to convert a molecule already in the blood made by the liver. Boy, that's not shocking. We already know already that the liver makes most of your blood proteins. And so this angiotensinogen is already in your blood made by the liver, and the renin is going to convert it to angiotensin 1. Angiotensin 1 is not the active molecule. It's still going to circulate through the blood, and when it gets to the lungs amazingly make this enzyme called ACE. And that ACE is now going to convert the angiotensinogen 1 into an even smaller molecule called angiotensin 2. Now, angiotensin 2 does three very important things. Number one, it signals your hypothalamus to tell you to drink. Oh my goodness, this sounds like lecture 1. Remember, we talked about water levels and the hypothalamus telling you to drink, and fluid levels, and fluid levels sounds like the kidney, right? We're coming full circle here. So the hypothalamus is triggered by angiotensin 2 to get you to drink. Well, we know when we drink, we raise our blood pressure. It's also going to go to your cardiovascular system and cause many of your vessels to vasoconstrict. If your vessels vasoconstrict, what happens to the pressure in them? It goes up. And angiotensin 2 goes to the goes to the adrenal gland and tells the adrenal glands to make even more aldosterone. Now you'll hear aldosterone, and you'll hear it pronounced aldosterone. I say aldosterone. Now, aldosterone is the hormone that is going to help you retain water and sodium. Okay, so what happens to your blood pressure when you retain water and retain sodium? Blood pressure goes up. Okay? So if you give someone an ACE inhibitor, you're blocking the ability of the body to do all of these things, to raise the pressure even higher. Okay, so you kind of take the kidney and the RAA system out of the individual's arsenal to increase their pressure. Their pressure is already high enough. That's why they're on an ACE inhibitor. So angiotensin II is a very, very potent vasoconstrictor. Again, when you hear about vasoconstrictors, you're thinking, oh, those things raise the blood pressure. Now, how do they do it? Watch this. They constrict the efferent arteriole. Okay, wait a second. They're going to squeeze down on the efferent arteriole. What's going to happen now? I'm squeezing on the exit. So that means blood is still coming in the afferent side, right? But I'm squeezing the exit door. It can't leave. So what's going to happen to the amount of filtration? It's going to go up. And as a result, you're going to make more, right? You're going to make more urine, right? So you're going to raise up the GFR even though you have a low blood pressure. So this is a great mechanism. It's going to help to kind of get around this whole thing. It also, as I mentioned in the picture, angiotensin II is stimulating the adrenal gland to secrete aldosterone, and that's all about keeping sodium and water in your body. And finally, I didn't say ADH a moment ago, but it's going to the hypothalamus, and it's going to release more ADH. And we know ADH promotes water reabsorption. So if you wanted to, you could go back here to this picture. I know it's small, but don't forget that this whole hypothalamus deal is about ADH being increased. So those are the three mechanisms: autoregulation, nervous system, and hormonal system that the kidneys are involved with, right? Myogenic and glomerular feedback, autoregulation, right? We need a lot of mitochondria because some of this reabsorption is active transport. I mean, the body takes a lot of energy to reabsorb these nutrients. This is not all by diffusion. This is not all passive movement. So the body actually has to expend energy to reabsorb many of these molecules. At rest, 6% of your ATP and calorie consumption is being used up by your kidneys simply reabsorbing the molecules that it wants to recapture. Right? So couch potatoes, 6% of the ATP, just sitting there, your kidneys are doing their job. Again, reabsorption is the reclaiming of water, the reclaiming of solutes. And where does it go? When I say it goes back into the blood, how's it getting there, folks? When I show this arrow, when I show this arrow going from the tube and things being reabsorbed back into the blood, where is it being reabsorbed? It's being reabsorbed into the peritubular capillaries. Remember, 85% of your nephrons have that tubing and have all those capillaries wrapped around that tubing. How do molecules get from the tube back into your blood? Not hard to imagine. There is transcellular. What does that mean? Going through the cell. Pretty regulated, right? If something has to go through a cell, it had to be welcomed in and escorted through the cell. So there's transcellular. There's also going to be paracellular, which means what? Going next to or basically around the cells. This would mean that molecules are sort of squiggling in between the leaky cells. So you've got molecules being reabsorbed both through transcellular mechanisms and by squiggling, leaking through and between these tubes. And there's this idea of solvent drag. That is, as water is moved, other molecules move with it, right? So you've always heard solutes suck. But it's also true that solutes would follow water. They're being solvent drag is pulling molecules, or water is being carried with the dissolved solutes. Again, all of this stuff is being taken up by the peritubular capillaries. What's running the show the most important molecule, and you thought you were done talking about it, sodium. We've been talking about sodium forever, haven't we? Action potentials, we talked about it in the muscles, and here we are again talking about sodium again. Sodium reabsorption is the key. Sodium, recall, is the number one electrolyte where? Outside your cells, aka extracellular, aka plasma, right? You agree plasma is extracellular? So the number one electrolyte in your plasma is going to be sodium. Sodium is going to be the major thing that is being filtered through, and sodium is going to run the show here when it comes to understanding what molecules are going to be moving back and forth. Sodium is the key molecule for creating both an osmotic and an electrical gradient. Okay, so osmotic and electrical gradients. We'll come back and look at that in a picture in a moment. And then there's two types of transport that are going to be moving sodium out, or I should say uptake. There's symports and antiports. Symports are going to be sodium moving from the fluid into the blood, but carrying something else. So not only is sodium moving, but it's going to carry with it a glucose or an amino acid or a lactate molecule. That would be symport. They're going together, right? Sym means together. Antiport is things moving in the opposite direction. So the sodium is moving in one direction, but something's moving in the opposite direction, and there is a sodium hydrogen antiporter. So that every sodium that gets pulled into a cell, acid, H+, protons, are going to be pushed out of the cell. There's also the constant sodium-potassium pump going on. Remember the sodium-potassium pump is what maintains the sodium and potassium levels in their rightful place. Without the sodium-potassium pump, you wouldn't have the right sodium-potassium concentrations to allow for resting membrane potentials and allow for action potentials to occur. So there's going to be, sodium is prevented from accumulating in epithelial cells by that sodium-potassium pump, which remember is always pumping sodium out of the cell. This is an active transport mechanism and does require energy. Remember too that wherever sodium goes, chloride always follows. So as sodium moves, negatively charged Cl- will always go with it. Let me show you this picture, then we'll pick up right here with this on Thursday. So we're moving, this is reabsorption. We're going from the tube into the blood, right? Back into the blood of the peritubular capillary. So what I'm saying is that there are things like sodium that are going to go through the cell transcellularly, and there's going to be some that's going to squiggle between the cells paracellularly. How the molecules are going to move, again, glucose and chloride and water, all of these molecules are moving from the tube over into the blood because of this idea of reabsorption. Okay, and that's where I'll leave it there for us today, and then we'll pick up with the rest of reabsorption, secretion, the last little bit of the urinary system on Thursday.